Yeah, so what we do in the lab is we use an in vitro batch fermentation approach where we could take human fecal samples derived from different compositions, and we could screen nutrients using this type of in vitro batch approach. From there, we do a bunch of different analyses, whether we sequence the DNA to get a good idea of where these compositions are shifting, but at the same time, you could spin down the plate, remove the supernatant and filter it, and then we could run it on a GC to get a good idea of the metabolic output that these communities put out in response to glycoproteins and carbohydrates. And we could actually use this method to screen other fibers as well, pectins, soluble fibers, prebiotics such as inulin, and use that as a good method to screen compounds.
That makes sense. Would you be able to talk about why GCMS is the right type of analysis for you?
Yeah, so traditionally with volatile molecules such as short chain fatty acids, since they're volatile, GCMS seems to be the standard to go for analyzing short chain fatty acids. The literature also uses, I think there's new methods. You could use HPLC, liquid chromatography as well. They're trying to move toward a type of assay where they have some sort of conjugated, not antibody per se, but something that could bind to short chain fatty acids, but the sensitivity and the accuracy of this type of assays aren't great. So traditionally, it's been GCMS or in some cases HPLC.